All right. Good afternoon, all. I'm Rekha. I'm a fourth year PhD student from the Stony Brook University. Without wasting time, let's get into the talk. Okay, so human genome consists of about 98% of non-coding regions. Yet these regions contain functionally significant elements like cis-regulatory elements, which play important role in regulating transcription through long-range interactions. Enhancers are one of these cis-regulatory element which increases the transcription of a target gene while interacting with their target promoters with the assistance of proteins like transcription factors, mediators, and RNA polymerase, thereby shaping the characteristics and functions of cells, organoids, and tissues. Any disruption in the ideal function of an enhancer due to genetic or epigenetic changes can lead to certain disease conditions. So, genome-wide association studies are a comprehensive repository of such SNPs which are associated with some traits or diseases. As of 2024, the catalog has a vast collection of about 625,000 significant SNP trait associations, which are linked to about 15,000 traits. Over 90% of these reported SNPs are in the non-coding region, and only a small portion of it has been identified as regulatory region variants. It is yet to unravel like how many of these are associated with enhancer regions, which are hidden in the large pool of these non-coding variants. This is mainly due to the challenges in enhancer biology, because we don't know, like it is unclear like the enhancers which gene it targets or in what tissue. Additionally, enhancers have these intricate characteristics which add another layer of complexity that makes the computational identification of these regions much more challenging. Most machine learning methods which have developed over these years have spotlighted the importance of extracting nucleotide a composition of enhancer regions. Though these models were good, but they failed to learn the actual sequence conduct of this region and perform suboptimally. We hypothesize that by building a model which can actually understand the actual DNA context of these regions can will be helpful in unrevealing the clinical significance of enhancer regions. So we developed a fine-tuned model called DNA Bird Enhancer using the pre-trained DNA Bird model, a genome foundation model which successfully captures DNA's contextual and semantic information in human genome. In this study, we demonstrate that DNA Bird Enhancer captures the contextual informations of these enhancer regions from the sequence alone through attention mechanism. So the dataset we used for developing this model is the enhancers from the ENCODE screen CCRE collections. These enhancers are from 378 different experiments on various cell lines, tissues, primary cells, etc. Based on the data quantity and also to learn the line dependency, we created two data sets, namely ESD201 and ESD350, which contains enhancer regions of their respective length. And this positive set enhancers, along with some additional enhancer data from public databases, we created the negative set, which is the non-enhancer set from the human reference genome. Okay. So using these two datasets, we fine-tuned pre-trained DNA bird model. The models were initialized with the pre-trained DNA bird weights, and we optimized the model through hyperparameter tuning based on epochs, learning rates, weight decays, warm-up steps, etc. And the training was executed on 8 NVIDIA GPUs, leading to development of about 500 fine-tuned models for each dataset. And among them, we selected the models with the high performance and with minimum training loss. So Now we have two models, namely DNA bird enhancer 201 and DNA bird enhancer 350. Both these models precisely predicts the enhancer regions with good performance matrices, where the DNA bird enhancer 350 with an accuracy of 88% performed better compared to the DNA bird enhancer 201, which has an accuracy of 82%. This indicates that the longer enhancers contain more regulatory informations, thus providing more sequence context than the shorter regions. Next, we validated our models by comparing it to some of the traditional baseline classifiers, and we observed that this DNA bird enhancer models outperform the baseline classifiers models trained on their respective data, followed by random forest, SVC, and Adabus classifier. When you look into the graphs, you see that the KNN classifier had a higher precision and a lower recall, indicating that the model overlooks or misses out the true positive values when predicting the high confidence enhancer regions. Our model also outperformed some of the recently published state-of-the-art methods from the performance matrices like accuracy, F1 score, and the prominent MCC scores, and also by observing the AUC and AUC and the PR curve. It is clear that our model shows superior performance in discriminating enhancer from the non-enhancer regions, followed by I-enhancer ELM, I-enhancer ECNN, and I-enhancer DCLA, regardless of the length of the sequences. These prediction results from both the baseline classifiers as well as the state-of-the-art methods once again prove that the models on 350 base pair perform better compared to the 210 base pairs. Next, we evaluated the genome-wide efficacy of our best model by identifying the enhancers in whole human genome. Our model predicted around 1.8 million. Our model predicted around 1.8 million enhancers of varying length from as short as 150 base pair to regions greater than 30 kb. We validated this result using the enhancers available in the public databases, and with a greater than 25% of overlap between the predicted regions and the publicly available enhancer regions, we observed that approximately 99% of our prediction intersects with the enhancers from at least one of these databases. Our model was also able to predict a greater portion of the enhancers publicly available out there, and prominently can be seen from VISTA, enhancer atlas, disease enhancers, and typical enhancers from the SCDB database as well. This suggesting that our model's ability to capture the potential enhancers beyond its training data. So identification of enhancers with varying length was possible by merging the consecutive predicted enhancer regions. Interestingly, we observed that these long regions overlap with different or multiple enhancer regions reported in the open source databases. One such region is depicted here in the chromosome 4. The length of this region is 42,450 base pair, and it maps to varying length enhancer regions reported in 5 different data sources, as you can see below. And this evidence further signifies the remarkable capability of this model to recognize and capture the enhancer regions of varying length through attention mechanism. Now, owing to the clinical significance we discussed in the earlier introduction slides, we applied the DNA bird model to predict the effect of the variance in this enhancer function. And using the dbSNP variants, we identified like 35,447 SNPs, which are significant to cause a loss of enhancer function based on the score change and the log odds ratio we calculated from the prediction probability from the model. Further, to investigate whether any of these variants are affecting or causing any disruption in the transcription factor binding sites within these enhancer regions, we used deep V regulome, which is a fine-tuned DNA bird models for 700 transcription factors and histone marks. So if anyone is interested in knowing more about the deep regulome, please attend a talk by Dr. Pratik Datta in the special session, which is going to happen in another 15 minutes. So, I'm sorry, I'm not able to find the mouse pointer. I'm not able to find the mouse pointer.
Oh, I see.
Oh, okay.
Here, let me give you a laser pointer here so you can see real bright.
Oh, okay. Thank you. Okay. So going back to the talk, so using this deep V regulome, we identified that about 43% of this loss of function variants is actually disrupting the transcription factor binding sites within these enhancer regions. And subsequent evaluation to find which any of these SNPs have any reported trait or phenotype associated with them, we queried and analyzed EQTL, ClinVar, and GWAS data and identified some interesting results. So highlighting here is the ISM plot for an intronic variant in chromosome 19, which causes a loss of function in the enhancer region, and it disrupts the transcription factor binding site for a protein called MAX in the within this enhancer region. On querying it in the GWAS, we see that this variant is mapped to a gene called PCAT19 and is also reported as a risk variant to prostate cancer. And in the UQTL analysis, it further confirms this, that this variant is reported to influence the expression of this same gene in multiple tissues, including testes. All these candidate variants, which are predicted by our model, are in or around the high attention regions learned by these models. So in this heat map or ISM plot over here, you can see the reference attention here. And the attention is mainly focused on this part of the enhancer region. And the variant is present over here, near to that attention region. When the mutation is being imputed, you can see that the whole attention of this wild type changed in the mutation nucleotide. We believe that all predicted variants present in this study provide a strong candidate for further experimental testing to confirm their functional impact on a specific disease or trait. So, concluding, DNA-BERT enhancer, it precisely locates and predicts enhancer region while outperforming recent state-of-the-art methods. And we identified about 1.8 million enhancer regions in the whole human genome by focusing on the high attention regions in the enhancer sequence learned by the model. Approximately 99% of these predicted regions map to at least one of the open source databases as part of the validation process. And it also identified around 35,000 unique candidate SNPs, which causes a loss of function in these enhancer regions, and out of which 15 of them have a disruptive effect on their binding sites. And further analysis for UQTL and ClinVar and GWAS also identified some significant candidate variants. I would like to thank GLBIO for giving me the opportunity to present my work here, also the NIH funding and my whole lab, especially my PI, Dr. Ramna Davludi, and my commenter, Dr. Farhatai, and the senior research associate which helped me with this study, Dr. Pratik Datta. Our paper is under review, and if you are interested, you can get it from the preprint copy in bioRxiv. GitHub page is also available for this, but we will be releasing the models only after the publications. Thank you all for attending the talk. Any questions?